All right, we're going to continue on our theme of restoration and restoration science and things like that. We're going to have our first speaker today is Jordan Teicher out of the Missouri Botanical Garden.
Thank you so much. Luckily, you have dead plants to excite you and reinvigorate you after your break. So really, Herbaria are experiencing quite an exciting time at the moment. It's been called a revolution, it's been called a renaissance, but however you decide to describe it, uh-oh, it's begun. There I am. I didn't do it. I don't think I did it. So yes, the reinvigoration or the renaissance or the revolution of Herbaria is tied mostly to two things. So new technologies and techniques that are enhancing traditional uses of Herbaria, which are still highly relevant, as we've seen from several talks at this conference, as well as new applications that were not anticipated when the collections were made. For this paper from Chuck Davis highlights some of the changes in those research applications. So these are new ones, including biodiversity, informatics, hydrogeography, global change biology. And you can see this rapidly rising portion of studies that involve herbarium specimens. So in terms of the traditional applications, we kind of already covered this in several talks, which is great. And whether you're conducting a traditional flora, whether you're trying to put together a world flora online, whether you're doing conservation red listing, conservation requires taxonomy, and taxonomy requires herbarium. That's where the crucible of taxonomy, identifying new species. If you ever try to describe a new species, trying to do it on the fly in the field with fresh materials is very, very difficult. Having that reference material, having libraries that typically accompany them is essential. So that role has continued to be as essential as ever. But digitization has made access to type material, heritage material, much easier. So we have GBIF with millions of specimens that can be accessed for that purpose as well. We're not going much faster as a result of this. I'm not quite sure why that is. There's a few different theories as to why it might be, but it looks like we have between 12 and 36 years, depending on how many we think we don't, how many we don't know. It's a hard number to estimate. Another application relevant to conservation, although a little bit less obviously directly maybe, is constructing the tree of life. So having a resolved phylogeny for plants gives us all kinds of insights that are useful for conservation. One is, as I think Gunther mentioned in his talk, one kind of diversity, phylogenetic diversity. And we want to preserve what we call evolutionary distinct units. Having this resolved tree of life is a way of doing that. Estimating what are we really at risk of losing in terms of really unique lineages that may have lost all of their kin. And then also there's a predictive value in knowing the evolutionary relatedness of plants. So if you're trying to do restoration applications, trying to preserve that kind of diversity, knowing that things are closely related to one another, that they evolve in similar habitats, can give you insights into what might be their responses to different biological threats. And the ability to get usable DNA out of herbarium specimens, mostly developed sequencing technologies, has been really transformative for herbarium in this context. So 28% of the samples from the first round of Q's Pastel project were from herbarium specimens. And I imagine that a larger portion perhaps of the second round as they try to piece together the genera that aren't represented mostly. So all of those millions of digitized specimens have also very clever people figured out that you could take those data and run them through complicated models, and you could make certain predictions about species distributions, better understand how species are responding to things like climate change, how their distribution might have changed in the past, how they might change in the future. This is an example that I really like, although it's a few years old. Wheeler et al. Invasion potential in different South African species. And so they did some climate modeling, took a bunch of specimen data of South African species like this ice plant, Carpodotus edulis, and got a climatic profile for it, projected, all right, if you have a climatic profile similar to this, where would you like to grow? And then mapped that around areas of the world and compared, is it growing there? And so that does two things. Well, one, it helps validate the model, say, yeah, it's growing where we expected it to grow. But two, in areas where it's not currently growing, but the climatic profile matches where it grows elsewhere, those are potential targets for conservation and for trying to prevent spread of invasive species. Spread's really aggressive, so it's all across California where it's not native. I used to have a really cute picture of my nephew picking it in California. It's a big picture. Distraction from the message. And the point I wanted, so I also highlighted some of the database composition, but the point I wanted to make here, kind of going back to that traditional purpose of herbarium specimens, is the distinction between a primary database and an aggregate database. So Tropicos and Sabif, South Africa Biodiversity Conservation Facility, they have smaller numbers of records, but these are really where the primary data are getting loaded into, where if there's any kind of curatorial updates, specimens are being identified, names are being brought up to speed. They're typically happening at those levels, and then they're being passed to the aggregators. So the aggregators are serving a really valuable purpose. It's an easy place to download tens of millions or hundreds of millions of records, but the bulk of the curatorial effort is often happening in primary databases that are pushing those data to those aggregators. So we shouldn't look past the sort of smaller, in terms of specimen number efforts, because that's really where valuable data are coming from. And that, after all, is what distinguishes data based on preserved specimens from data based on observations. That verifiability and that updatability, where you can go back after the fact, look at the specimens again, update the names accordingly, and keep that on the update. I'll return to that. So other unanticipated applications. So extraction of functional traits. Perez et al. Paper in 2020 found they looked at 3 different functional traits useful in ecological performance. In specific peak area with specific gravity, volume of the branches, and leaf thickness, and found they could be consistently estimated from dried material on various specimens. You see these lines don't necessarily align exactly with one. Exactly aligning with the dotted line there would mean that it's a one-to-one. They're slightly off, so you might have to correct slightly for the way that plants dry. But overall, you can make realistic estimates, accurate estimates from herbarium specimens, which opens up huge potential data resource. Even more rapidly, some of those traits can be estimated automatically through machine learning approaches. So the computers are getting pretty good at looking at a specimen and saying, oh, this is a label, and this is a leaf, this is a stem, this is a flower. So you can imagine, as we're digitizing millions of specimens and creating these high-resolution specimen images, if you can run over the course of several weeks a machine learning approach and extract all of these data, still have them linked to the specimen records, you can start accumulating a really massive data set of ecologically relevant. Multispectral analyses, another way that we could potentially rapidly extract data from specimens. So there have been some preliminary studies showing that there's a phylogenetic signal that shine a multispectral spectrophotometer on living tissue, is this example. It shows some level of phylogenetic distinctness. Sari et al. Extended that to dried material and looking at functional traits as well as taxon ID. So in this limited context, found that, so this is when you see 100%, that means 100% of the specimens were accurately identified based just on a multispectral profile and a discriminant analysis. Now that's out of a subset of species that were in this analysis. So presumably, we would anticipate as you expand the analysis dramatically, you're going to run into fewer successes. But still, you could get them to maybe major clades, major families, major groups, and some of these can be applied very rapidly, either herbarium specimens, creating that fingerprint, or in the context of maybe a flyover. So those were mostly applications that make it more convenient. So herbarium specimens are easier to get your hands on. It doesn't involve difficult and expensive field work. But there's also unique applications that you can only do with various specimens, and they are basically the only physical record of the past 300 years of plants we are likely to ever have, or at least the very best. And so the ability to get DNA out of herbarium specimens also represents a little snapshot from the past. Same thing would go for things like isotopes, chemical profiles. Really the only way we can learn about what plants we're experiencing that are no longer with us. So this is a great study that I really love. Again, a little bit of an older one, but Kristen Saltonstall did some genetic analyses for invasive genotypes, native genotypes of Phragmites. If you're not familiar with Phragmites, if you have them next to one another, the invasive and the native look quite distinct. But if you're a land manager and you've just encountered one of them, and you need to know, is this the native North American genotype or the invasive? It's very hard to tell. And she was able to use herbarium records going back prior to 1910 to track the invasion of that non-native genotype through New England and then spreading throughout the United States, where now it's the predominant one. If you run into one, odds are good that it's an invasive genotype. And also found that there were at least 2 native genotypes. So this is really important for preserving, trying to conserve native species or do reintroduction programs. You may need to reintroduce Phragmites australis into a population of Phragmites australis, right? Because you have an invasive genotype that's quite aggressive, clogs up waterways. You have a native genotype that does not do those things, but also seems to prevent the invasive genotype from invading. So where you have native Phragmites, it tends to not get invaded by the invasive. It's where you have sort of clear-cut wet areas that you get invasive. And then as a, it's a pretty low success rate, but they can also, herbarium specimens can also be sources of viable germplasm in rare cases. So rare pieces of ferns, as well as Hawaiian plants. If you have no other course of action, if it hasn't been seed banked, at least potentially worth a try. Look at the herbarium specimens. It really depends on factors of that are taxon specific. It depends on what the storage conditions were. Herbaria are not really the same in storage conditions as seed banks, especially the way that the plants are dried and initially processed, especially the way that they were processed historically. But we get some small amount of germination from some species. So thinking about the future and how are we going to collect for these new applications? All of them so far are things we didn't intend. They were collected mostly for taxonomy, were collected as vouchers for individual projects. And it's in the aggregate with these new unexpected technologies that we found these. One argument that's been advanced for including them in ecological research is that biodiversity research without vouchers is not verifiable. This is technically true for a lot of different cases. It's not particularly sexy. It's not particularly inspiring. And so the way I would like to think about it more is that they offer the potential for an ever-growing, sort of self-sustaining body of knowledge in a way that our other research typically doesn't. So because they all tie back to a physical, verifiable specimen, if we can get the data organization and intercommunication right, you have an ever-growing body of information, of data, all connected back to the same organism. So for example, if you go out and measure an interesting trait like a specific leaf area, and then you measure another trait to model density, and you want to know if they're related, you have to measure those both at the same time. Versus if we've been recording all the things we've ever measured on herbarium specimens since we started measuring on herbarium specimens, we would have tens of millions or hundreds of millions of data points connected to single organisms that are consistently, continuously updated in terms of their curation. So the best identification on them, a verifiable identification, and then an ever-growing body of interconnected information, which I think will be even more exciting and relevant as our ability to write models, really realistically for computers to write models for us that are increasingly complex. So right now it might be difficult to wrap your head around how would you analyze all of that interconnected data, but that's not far away. There are certain challenges. So we have this slight paradox of it's a really exciting time for Herbaria, but they're also being closed frequently. The reason for this, well, there's a few reasons, but I think one of them is the new applications that are exciting for the scientific community are typically realized in the aggregate, where you have millions of data points, they're scattered across multiple herbaria. If you look at these ecological papers, they'll often cite in the acknowledgement section a very long list of Herbarium acronyms if they cite Herbaria at all, or they might be downloading a giant download from Gmail. Wonderful, we love that our data are being used, but you can see if you're an administrator at an institution, that's not a particularly compelling case of some of our specimens were used along with 18 other institution specimens in this study. So they continue to close. Additionally, if we want to think about new applications, new collections, there's been a lot of proposals about how can we incorporate all of these fungi in the soil, how can we do DNA samples, can we collect seeds at the same time. Those often require special technology to count them, and they require expertise and staff to manage them. So the administrative burden also grows there. If we want to continue collecting, especially for ecology, my recommendation, or one idea, I don't want to come off that strongly, maybe just something to think about, but more regional collections, having smaller herbaria associated. I was really excited to see the herbaria at some of the smaller botanic gardens and the newer botanic gardens. It's really wonderful. That does create curatorial challenges of if you have fewer and fewer taxonomists for identifying all the specimens, and the specimens are more dispersed, well, then that real strength of having the specimens identified as accurately as we can is a bit harder to maintain. The future directions, I'll be quick because I think we're running out of time. We increase the number of regional herbaria. For example, as of 2011, maybe it's skyrocketed since then, but I haven't checked the update. Only about a quarter of botanic gardens had herbaria. There's massive potential there, but only if we sort of commit as a community to this idea of a shared resource that's primarily valuable in the aggregate. So it's not necessarily going to yield for you immediate large-scale financial benefits. And that's a tough thing to sell to administrators, but it has tremendous value collectively. Some of those costs can be offset by just being mindful about how we're writing our grants. We typically put every other cost into a grant proposal, aside from the processing costs of specimen, and we deliver it then saying, please take care of this forever with no money. That's very hard for Herbaria to maintain. And then critically, it's the better integration of databases. So we can do this now. We just kind of have to get our act together. Unique identifiers for specimen records, provide that link. So you should theoretically be able to very easily pull information from GenBank, get an updated identification for that from World Flora Online, which is feeding data back and forth between it and Tropicos, which is updating determinations that feed back into Paris because they have duplicates of those specimens. None of it works that way. GenBank has to be edited by hand. Paris has to be edited by hand. All of them have to be edited by hand. I'm going to just put these up because I think I'm out of time. But thank you very much for your time, and I'm happy to answer questions.
Okay, I do believe we have time for at least one quick question.
Can you be reached for consultation?
Any other questions? Okay, we're continuing on. Thank you.